My project this summer is entitled The Experimental Study of the Ultrafast Transient Optical Response of Gold Nanoparticles by Broadband Laser Spectroscopy. Light incident on a metal nanoparticle causes the conduction electrons to oscillate. Excitation of the nanoparticles at a resonant frequency results in the creation of a strong electromagnetic field. This phenomenon is known as localized surface plasmon resonance, or LSPR. Thanks to the properties of LSPR, one can quickly and efficiently inject energy into the nanoparticle system using ultra-short pulses of light. It has been shown that through the excitation of gold nanoparticles in water, it is possible to generate reactive oxygen species, or ROS, such as the singlet oxygen, superoxide, and hydrogen peroxide. High concentrations of ROS can lead to the death of cancer cells. Our lab seeks to establish a link between a generation of ROS and the presence of electrons in solution. The first step in my project was to use pump probe spectroscopy to detect the solvated or free electrons in water. In order to increase our chances of detecting these electrons, I assembled an apparatus to sparse the dissolved oxygen inside a sample of water by flushing argon through the system. A pump circulates the water and argon while a balloon catches the excess oxygen eliminated from the circulation. The sample is then irradiated by the pump probe setup. This setup consists of splitting a laser into two parts. The pump beam passes through this topaz, which allows us to change the wavelength of the output light. The probe beam is transformed into white light by focusing it on a sapphire plate. The two beams collide inside the sample. The pump excites the sample and the probe measures the absorbance. After detection of solvated electrons, our lab will excite gold nanorods of varying aspect ratios to optimize the production of reactive oxygen species. We expect to observe a directly proportional relationship between the generation of solvated electrons and the production of ROS. Ultimately, we hope to localize the production of ROS to kill cancer cells while keeping healthy cells unharmed.